And now what we're going to do is move from climate and weather science to biology. And to kick us off in that regard is Anshul Kundaje, is that right? Kundaje, who is an assistant professor of both genetics and computer science. And so he's going to bring those together and tell us about deep learning for genomic discovery.
All right, let me just make sure I turn my timer on so I don't run over. Okay, so thanks so much for the invitation to this great conference, and it's been fascinating listening to applications of AI in all kinds of domains. So I'm going to switch gears and talk a little bit about applying machine learning, specifically deep learning, to understand the human genome and genetic variation. And it's sort of continuing the theme of using black box machine learning methods, but applying various kinds of interpretation approaches to really focus on discovery versus just prediction. So I'm going to start by motivating, just looking at diversity in the human population. There's incredible diversity in the human population in terms of all kinds of benign human traits and phenotypes. For example, various attributes. For example, the type of hair that you have, the height, various other kinds of traits. But there's also variation in susceptibility to various kinds of diseases. And a lot of this variation that we see in phenotypes, human phenotypes, can be attributed to actually genetic variation. So these are, substitutions, insertions, deletions in our genome at various positions. And these can influence a whole variety of different benign traits as well as diseases such as cardiovascular disease, type 2 diabetes, and so forth. And so there's been a lot of effort to actually catalog genetic variation in the human population. And so I'm going to focus on sort of the two key axes of large-scale data available in genomics. Since the Human Genome Project finished its first draft in 2003, there has been an explosion in revolution in genome sequencing technology, high throughput DNA sequencing technology, and that has resulted in a super exponential increase in the number of human genomes being sequenced, and a corresponding drop in sequencing costs faster than Moore's law. The sequencing machines have also, as you might expect, dramatically reduced in size. You now have these USB-sized machines you can plug into your laptop and stream sequences into them. And that's a population scale sequencing. So there are countries that are being able to sequence as part of their public healthcare system. You can opt in or opt out, but they can sequence the entire population. And so when you do that, you can essentially catalog all these different positions in the genome where our sequences are not identical, right? And that's referred to as genetic variation. And there's obviously great interest in just cataloging this variation because it gives us so much interesting information about the genetic basis of diversity in humans. But you can do other interesting things with this data. You can do case control studies where you not only sequence individuals that are relatively, in quotes, healthy, but you can contrast their genomes or their genetic variation against a bunch of other individuals that are inflicted with a specific disease, for example. And this is commonly referred to as genome-wide association studies. And the idea is, again, you got summary statistics of genetic variants, which we also observed in the two classes, cases versus controls. It doesn't have to be binary. You can also have continuous traits, for example, height or blood pressure. But the key point is you can take each of these genetic variants and statistically associate them with variation in the phenotype you're interested in. And you can identify certain variants that are highly associated statistically with the phenotype of interest. So to give an example of what this looks like, let's say I'm looking at Alzheimer's disease, right? And I've done a case control study contrasting genomes of individuals with Alzheimer's versus healthy controls. This is a Manhattan plot, as you might imagine why. It looks like the skyliner of Manhattan. The x-axis is the entire genome, 3 billion base pairs, human genome. Each point is a genetic variant in the genome, and the y-axis is plotting the negative log 10 of the statistical significance of the association of each variant with that disease. And you can see most of the genetic variants are below that line, which is the sort of threshold for significance, but then a whole bunch that really skyrocket, right? So these are loci in the genome that are genetically associated with susceptibility or risk for Alzheimer's. You can do this for hundreds and thousands of diseases and traits, and thereby start mapping all the loci in the genome that are associated with disease. But this is a great way to detect loci. But then the next question arises: well, okay, if I found this A in the genome and there's a mutation at that location, let's say the A was transformed into a G in individuals with a specific disease. So what? Now, what do I do with this? So what I showed you before is a detection approach to detect variants associated with disease. We now care about the functional consequences of that variation. And so we're going to start talking about functional consequences of variation. Now we're going to start understanding the genome, right? So I'm showing you sort of a cartoon version of what the genome looks like. There's the cell, the nucleus, unspooling the DNA, and the DNA contains various kinds of functional elements. Everyone knows of genes. There are about 26,000 genes in the genome. They code for proteins. You can directly read the sequence of the gene, and it translates to an exact protein. Now, what defines cell state and cell identity is the specific repertoire of proteins that are expressed in that cell. And so, for example, there are genes that are active and expressed, and there are genes that are repressed, and the specific cocktail of genes that are expressed gives rise to that specific cell state. So if you're activating and repressing genes, you clearly need a control system that turns these genes on and off. And what's really interesting is the control system of gene regulation or gene expression is also in fact encoded in the genome. So I'm showing you these little yellow boxes. Think of them like on-off switches. They're control elements that turn genes on and off. And the way they function is they act as landing pads for various proteins, DNA binding proteins. These are often called transcription factors. These DNA binding proteins are about 6,000 of them, and they operate in complex combinatorial fashions, and they act, and these control elements act as landing pads for these proteins. Now, what's really interesting is evolution has designed these proteins to recognize very specific words and syntax and grammar that's embedded in these control elements. So there is another language that actually controls gene regulation. It's a fuzzy language. It's not like a classic like natural language where you have discrete words and so forth. But it controls affinities of proteins to DNA. It controls the combinatorial interactions that proteins have with each other and with DNA. And then the specific combinations of proteins that land on these control elements and the specific constellation of elements that are active in a specific cell state give rise to the gene expression programs that define cell state and identity. And so why is it relevant for disease? Because as I mentioned before, if you take all these these huge maps of genetic variation and the association with disease, and you think about how these genetic variants are working. So for example, let's say there's a position in the genome where you have a C at one position, in healthy individuals, and the risk version of that variant is a gene. So how might this variant be functionally affecting the disease phenotype? So broadly, there are two ways in which this can happen. The genetic variant can directly break the sequence of the gene, right? So the variant is inside the coding portion of the gene, and if you break the sequence of the gene, you messed up the protein sequence itself, which is going to disrupt protein structure and function. These are often called coding variants. And the other way you can do this is, and of course, you might imagine those kinds of variants having very catastrophic effects. If you really destroy the structure of the protein, you're going to have probably very severe diseases, including potentially fatal outcomes. But another contrasting strategy that variants might affect phenotype is through control elements. So you're not disrupting the gene itself, the gene is intact, but you're changing the control system that precisely regulates which gene is active in which cell type at which time point. So you can imagine in development, you have this very precise program of genes that are expressed, and you have mutations in the control elements, and they start turning things haywire. Right? And what's really interesting is if you take all the genome-wide association studies that have been done to date for hundreds and thousands of diseases and traits, and you look at where those variants actually lie, 95% of them do not break genes themselves, but they in fact break the control elements. And this makes sense because we typically get diseases like obesity, Alzheimer's, type 2 diabetes much later in life, right? And they're not catastrophic the day you're born. And so that's happening through disrupting the control system of genome. All right, so if you want to start mapping where these control elements lie in various kinds of cell types and cell states, you're going to need some data, right? I'm going to get to the machine learning part of it. But I talked about the large scale genetic variation data. Now we need a sort of molecular mapping data to identify all these different proteins, where they bind in the genome and which cell type and what condition. And so thankfully, again, the genome sequencing revolution has enabled also a revolution in molecular profiling technologies. And so what you can now do is, if you're interested in, for example, this protein, this black triangular protein, and its binding locations in the genome, you can perform one high throughput sequencing experiment. What that sequencing experiment does, it uses clever enzymatic tricks to capture sort of the molecular shadows or footprints of these proteins. So you end up with one experiment that gives you like a track across the entire genome with some signal, and the signal peaks are the likely positions of where these proteins bind. And you can not just do this for one protein, you can do this for hundreds of different proteins and all kinds of various molecular markers of function. And through an experimental strategy, you can actually start profiling the entire genome and getting all these different molecular measurements, right? Now, what's really interesting is if you take all the cell types and tissues in the human body, they have the exact same genome, right? They have the exact same genome. And so what's actually happening is you have the exact same code in all the cells, but that code is being differentially translated into different kinds of functions. So cell state and cell context really matter in biology. And so what, sort of going back to the question that I asked the previous speakers, one of the challenges in genomics has always been the lack of massive standardized data sets that can be used for machine learning. So since the Human Genome Project ended in 2003, there are huge efforts funded by NIH and various other funding agencies. And what we now have is essentially this kind of, just as we have the reference genome, we now have sort of a reference molecular map of the genome. And that has three axes. You have the genome number axis, there's 3 billion positions. You got hundreds of different molecular readouts on the other axis, that's the second axis. So that includes like proteins, gene expression, all these molecular modifications. And then the third axis, the vertical axis, is actually infinite. It's all different kinds of cell states, cell types, tissues in every individual, right, in the human population. And now we'll be able to do this at the resolution of individual cells. So you have this incredibly massive molecular map now, and you're trying to make sense of it. All of it is somehow encoded in the genome. The 3 billion base pairs of the DNA encodes this giant tensor, right? And you want to learn a mapping from DNA sequence to this sort of molecular activity in all 3 dimensions, right? So that's where machine learning models come in. And I'm going to show you today some case studies of how we can use deep learning models, particularly to take these kinds of data sets, massive data sets, very high dimensional in multiple dimensions, and actually use them to try to learn fundamental biology of the object. And also how we can use the models as organisms to make predictions, causal predictions about correlations to the DNA sequence. That is through genetic variation. All right, so we start diving into the machine learning part now. So one of the different kinds of models people have built over many years. Since 2015, neural networks have kicked in as one of the powerful approaches to learn these mappings from DNA sequence to these molecular profiles across the entire genome. So think of these like text-to-speech converters. They're usually convolutional networks or transformers or whatever you have them. But the input to the model is DNA sequence, and the output of the model is a profile across the entire genome, and the goal is to learn a mapping from sequence to profiles. And these profiles are very context specific, right? It depends on the exact molecular event you're trying to measure, and it depends on the cell state or cell context that you're trying to measure. You can fit these models really well, as you might imagine. Neural networks are very powerful. But there's no real point in sort of fitting the model if you're not actually able to understand what it's doing, because we already have the data. The point of fitting the model is to learn the translation from sequence to activity. And so going back to some of the XAI aspects that the previous speaker talked about, we've also developed wide these different kinds of feature attribution methods. And the idea is once you fit in a neural network that can map sequence to activity, you now reverse engineer how the neural network is actually functioning. So let's say this model is taking in a sequence and able to very accurately predict this profile, molecular profile. You can backpropagate the output recursively through the model and get sort of contribution scores of individual bases in any DNA sequence. What are the contributions to the prediction made by the model? I just want to show you that first, sort of show that these models are in fact extremely accurate. The recent models we published 2 years ago called DPLab, they can take these molecular footprints of different proteins. These names we don't recognize. These are just names of proteins: OCT4, SOX2, NanoG, and TLF4, extremely famous proteins that are used to reprogram cells actually. And you're seeing the observed profiles and the predicted profiles for the 4 different proteins at 2 specific locations in the genome. There are thousands of these kinds of locations. And you can see the models make very, very accurate predictions at single base pair resolution. In fact, if we quantify them, the accuracy of the model is on par with replicate experiments. So we're able to really get very, very accurate mappings. What was more exciting is we can now start taking every element of the genome that seems to show molecular activity related to gene regulation, and we can start reverse engineering the bases, the contributions of individual bases that are driving binding or driving activity of these various proteins. So here I'm showing you one specific enhancer. It's a regulatory element that enhances expression of a gene called OCT4. All of these 4 proteins bind together cooperatively at that site. And they take models trained on each of these proteins individually. So you have 4 models. They're including the same sequence with the lens of 4 different models. You're capturing binding of these 4 different proteins. They highlight really beautifully sort of the complex syntax that's in the sequence. Because of the same sequence of differentially interpreted by different proteins, you have the center word that's common to all, and then you have this little TG80 word that's specific to this nano protein. And if you look at the flanks, these GGGG flanks, they're specifically affecting KLF4. And so we can start getting these really beautiful, very high resolution annotations of the DNA words that are trying to drive the binding of each of these proteins in every cell context. Now, you can do this for every element in the genome, right, in every context, but you might want to summarize the global lexicon, the recognition codes of these proteins across the entire genome. And so we have another approach called Morisco, and this is sort of a distilling approach. The idea is we can use these attribution methods to interpret every sequence in the genome, right, all 3 billion base pairs. These attribution scores, contribution scores that tell you which pieces are found. This is what the model and important words. And we do massive clustering algorithms on these words, and we can start aligning them, collapsing them, and starting to compress the millions of parameters into a compact set of lexicons, recognition lexicons. And it's actually quite fascinating. If you just focus on 4 proteins, okay, in one cell type, what I just showed you before, do the 4 proteins recognize just 4 words? The answer is actually no. The 4 proteins together recognize about 50 different words. And the reason is because they have incredible combinatorial cooperativity. And so there's all kinds of complex heterodynamics, like the first word that there's home minimums, there's all kinds of recognition codes. There's even interrecognition proteins by other proteins. And all of them produce beautiful, distinct shadows in the when they bind the genome. So we can really start tearing apart the very complex lexicon of binding recognition codes for these various proteins. But let's go one step further. We can go beyond lexicons. We can also go to syntax. There are various aspects of genomic syntax, one of which is the precise spacing and orientation of these words. Right? Just like in language, you care about which word comes before which word. All of those aspects make a difference. We can start using these models in silico oracles to actually dissect syntax out of them. And we use 2 simple strategies to do this. The first strategy is to design synthetic sequences where we insert specific words that we learn from the lexicon, and we can change certain properties. So, for example, let's say I inserted this yellow word and the orange word, and they bind different proteins. I can change the spacing between these 2 words. I can construct synthetic sequences where all the times inserted 2 words and I change the spacing, and I take what happens when I start changing the spacing between these words. The other approach is I can take actual genomic sequences which have words embedded in them. Evolution has embedded these words in them. And then I can start doing more differential experiments. I can mutate this word and I can say, what happens to the binding of the yellow protein if I mutate that word, right? So let me show you an example of what this looks like. This is a simulation where we train a model, and the model, there are two models here. One is predicting binding of the yellow protein, the other is predicting binding of the red protein. Each of them has their own recognition words, as shown right here, motif 1 and motif 2. I'm constructing a synthetic sequences where I've inserted the two words. What I'm doing is I'm changing the spacing. Okay, they initially 150 base pairs apart, and then I start moving them towards each other. And what you see at the bottom is actually an animation of what the model is predicting as the words move towards away from each other. What I can do is I can graph the binding, change in the binding of these two proteins as a function of distance. And that's what this looks like. So the x-axis is the distance between the two words, and the y-axis is the change in this in the binding strength of the two proteins, the red protein and the orange protein, as a function of distance. You see something really fascinating happening. The first thing you see is that the red protein doesn't care, right? So you can change the spacing between the two words, and the red protein is a flat line, like there's no change in its binding. If the other protein, the orange protein, the golden protein, you see something very different. As the two proteins approach each other, you see its exponential rise in binding. So there's a very strong cooperative interaction between the two proteins. But it's not just that, as the exponential rise happens, there is some interesting periodicity that's kicking in, right? And what's really cool is that periodicity is exactly 10.5 base pairs, which happens to be the helical tone of DNA. And what the neural is telling us is that these two proteins like to bind on the same side of the helix. When they do, at multiples of 10.5 base pairs on the same side of the helix, they cooperate. If they bind on the opposite side of the helix, they do not. So this training on the black box model, just training data agnostically, is able to learn some very fundamental principles about how proteins bind DNA. It's able to learn the helical, the cooperative effects, and so forth. Again, this exact experiment again conventionally in the genome. This is again back to the sequence I showed you before. The actual enhancer in the genome. There's this OXOX1 and now we can sequentially start mutating one to make predictions. So when you delete the OXOX1, binding of OX4 and nano both disappears. If you delete the nano word, you can see that it disrupts nano a little bit, but not so much OX4. And if you aggregate this relationship across the entire genome, you again recover the same sort of pattern we saw before. So the synthetic sequences are agreeing with the in-genome mutagenesis experiments that we're doing. So we've been applying this to thousands of proteins, thousands of pairs and triplets and higher order combinations of proteins, and we can really start tearing apart the very high resolution syntax, the lexicons and the syntax of the genome in hundreds of cell types and tissues. We can, of course, then design actual experiments downstream. So here I'm showing you, we can test millions of hypotheses in the model. And these experiments are really quite laborious and expensive and difficult to do. These are actual in-genome editing experiments using CRISPR technology. We can scan through hundreds and thousands of potential perturbations, look at ones that look interesting, and then prioritize those for experimental validation. And your juniors likely who's our collaborator of our lab actually perform these experiments. And if you just compare the top and the bottom, you can see that the observed and predicted match very, very nicely. So they actually validate the syntax the model learns at very high resolution. So let's go back to our, so this is sort of capturing, using perturbation strategies with models to discover, the basic syntax of the genome. But let's go back to our question of how we can use models to also predict effects of actual natural genetic variation. So you have to show an example of an actual genetic variation of the genome, which is in these kinds of downstream molecular events. There's a benign allele, which is a C, and then there are certain individuals that have a G. Okay, and we can use a model to predict what would happen if we mutated that sequence. We took the sequence and we switched that C to a G. You can see the model predicts that this massive amplification of binding, right? This is an actual genetic variant. These are not synthetic mutations that we are making, like in the previous case study. These are actual genetic variants we're looking at. We can go one step further and say, okay, so this genetic variant has a huge effect. How is it having that effect? We can again use our interpretation tools and take the sequence with the C and take the sequence with the G and look at the sequence attributions. And the model has a beautiful story that when there's a C right there, there's a weak DNA word on the left that's driving the weak blue profile. When the C switches to a G, it creates a word, a DNA word that induces very strong binding, and the two words combine to give rise to a very, very strong cooperative effect. And so this way we can start integrating how every genetic variant in the genome is influencing all these molecular activities. Let's go back to Alzheimer's case study, right? This genetic, this massive clock that I showed you before. But all the genetic variants that are associated with the disease, we can now start making sense of what they are. So to do that, we actually move to the brain. Obviously, Alzheimer's is a disease or neurodegenerative disease, so it affects the function of the brain. And the brain is an incredibly complex organ, and it has incredible diversity of cell types in them, cell states. So what we learned originally is very new technology that's emerged in the last year or two, single cell genomics. So all the measurements I showed you before, the molecular measurements that you can make them now at the resolution of individual cells. So the way you can do this is you can take the brain tissue, and in every individual cell, you can start measuring these molecular readouts of activity. What we can then do is take sort of the vector of activity for every cell and compute a cell-by-cell similarity matrix, right? And then we can discover heterogeneity in cell state space. So from this sort of cell-by-cell matrix, you can cluster cells and you can project them into a two-dimensional embedding, and you can discover more than 45 or 25 or 30 different cell types in the brain, right? Without knowing what those cells were, just by looking at molecular similarity of cells. And so we can discover all kinds of new types, oligonucleotides, microglia, the new type of cells in the brain, cells. What do you think? For each of those discovered cells, cell types and cell states, we can aggregate the signal, and we can essentially get pseudo profiles for every cell type. Okay, these molecular profiles that I showed you before. We can train neural networks mapping the sequence to the molecular profiles now in each of these different cell types in the brain. And then we can run them through this perturbational exercise. We can take every genetic variant in the Alzheimer's patients, and we can push them through the model to predict the effects on molecular activity in every cell type that's detectable in the brain. We're going to show you one example. This is a very important locus. This is a locus that has a very high association with Alzheimer's disease. There are about 165 genetic variants in that locus that are found in the population. When we apply the strategy through the lens of the model holds it on just a single variant, the one showing red right there, as the likely causal variant that's driving this locus. And as I showed you before, we can actually interpret what this variant is doing, and the model predicts, makes a very specific prediction that this genetic variant, RS blah blah blah, disrupts the sequence motif of a phosphoprotein in a control element of the pipeline gene in the oligodendrocyte cell types in the brain. Extremely specific predictions about the mechanism of action, the gene that it's affected, the cell type, and how it's actually affecting, right, through the binding of this protein phos. So we're able to map this way hundreds of GOS loci in Alzheimer's and various other diseases. But I want to show you that we can do this also for rare disease. So what I showed you before, Alzheimer's, obesity, type 2 diabetes, these are all complex diseases that affect vast swaths of the human population. There are rare diseases, for example, congenital heart disease. In this case, these diseases are very specific to specific individuals, and there's no statistical data that we can use to kind of bootstrap your model because it's an N1. There's only one individual who has that type of mutation. So how can we solve this? Again, the machine learning models come to the rescue. Here we perform molecular profiling in human fetal heart. This is sort of one of the first data sets going really early into human fetal heart development. And we take human fetal hearts at post-conception week 6, 8, and 19. And we again discover all the cell types in the heart, we profile them. We fit machine learning models that can map DNA sequence to the molecular profiles of all these different cell types and cell states in the human fetal heart. And then we can take case control studies again, but these are rare diseases. These are children who have congenital heart disease. We can take all their genetic variants, we can push them to the model, and the models can make these kinds of predictions that, there's again this mutation in a specific patient that disrupts the motif of the ETV protein active in arterial endothelial cells. So again, how many of the mechanism, the likely causal mutation, the likely gene, and the cell type of action. And again, we can drive downstream experiments. So our collaborators took these predictions and actually performed real experiments, again, CRISPR experiments. And you can see quite nicely that when you induce the mutation in the correct cell type, the experiments validate not just the molecular effect, but in fact, if you take stem cells and you induce the mutation in the stem cells and you differentiate them into arterial endothelial cells, patients, the cells that have the mutation, in fact, produce arterial endothelial cells with severe morphological disformation, right? So it's showing that there's a phenotypic effect even on the morphology of the cells in this way. We've done this also for autism patients, again, looking at human fetal brain, very similar kinds of predictions. So this can be applied as a general tool to start studying genetic variation in complex as well as rare diseases. And just to go back, one of the important things is to empower not just machine learning people and ourselves to be able to do the research, but to also help all kinds of other groups work with the models and use them in interesting ways. So in these applications of some machine learning, people have these models. These are repositories for models with convenient APIs. You can easily pull them down. Enables sharing of models, reproducibility, fine-tuning, transfer, all of these aspects. So in collaboration with a few other labs, we built one of the first models used for genomics. It's called KIPI, which is a Greek word for garden/zoo. And this is sort of a model that we will continue to build, trying to a whole variety of different models with very easy to use APIs. You can basically put models and use that in a matter of minutes versus having to reconstruct and figure out environments and so forth. And so using these strategies, we're hoping that we can democratize the use of these for interesting downstream applications. So with that, I'll just summarize and say, hopefully my talk today shows you that large-scale molecular profiling datasets and genome sequencing is really helping us decipher genome function. Scan map DNA sequence to molecular profiles with very high accuracy. The models can not like uninterpretable black boxes. You can actually query them to decipher functional DNA words, letters, and syntax. You can make really beautiful causal predictions, even asymmetric causal predictions. The models can be used to decipher common disease-associated genetic variants, and the models can be used for downstream experimental design, and many of the predictions actually validate very nicely in various kinds of genome perturbation experiments. And so, of course, I want to, of course, thank my lab, and also just mention that all of this work is super collaborative, part of often very large consortia like the ENCODE project, Roadmap of Genomics, and it's a very interactive effort where the machine learning models often drive experimental design. The experiments then drive the next phase of machine learning models, and sort of there's a nice positive feedback loop in the process. And with that, I'll just stop. Thank you.
Which is we talk about genotype.
You talked about genotype to binding. How good are we at going from binding to gene expression? Can you speak to that?
Yeah, I think we're sort of starting to move to the molecular chain. So binding is closest to what's happening to the proximal DNA sequence, and we're very good at that. We're good at various other kinds of proximal modifications. I think for gene expression and for many other molecular events that integrate information across the entire genome, you need models that can actually look at a much bigger receptor field. They need to model entire chromosomes often. And as soon as the people have tried this, there's a famous paper that came out last year that tried to do this by just throwing a giant transformer at it. It failed miserably because actually there isn't enough data to just like scale the models up. So one of the interesting things that we need to do is figure out, given the sparsity of information we have, even though it looks like it's huge data, it's contextually massive, but within each context, the amount of information you have is pretty sparse. So there are interesting efforts now in our lab and other labs trying to move up the molecular chain of events. So, from binding to gene expression to signaling pathways and eventually cellular response. It's, in my view, it's totally feasible. I think what's really important is to get the right kind of data in the right cell context, and then not just throw kitchen sink foundational model-like efforts. My personal view is in biology and genomics, the kitchen sink approach is highly flawed for many reasons. Not just excluding the fact that the data doesn't allow itself to be just thrown that way, but also the fact that we really want to use the models to interpret the genome. And as soon as you start developing kitchen sink models, we have evidence coming out soon that you have all kinds of feature leakage between tasks and all kinds of horrible things which you do not want them to happen at all if your goal is to use the models for motivational experiments or for design. You'll be extremely careful about how you design your models because data have a whole variety of different kinds of biases. And focusing more on data design, bias correction, in my view and in our experience, is far more powerful than just throwing the biggest model you can get at.
Yeah, sorry. Okay, so take a shot. There are people who
So this maybe goes to or relates to Aaron's question. There are some groups looking at things like high C data for chromosomes. Is there a pathway or a scope to sort of adapt what you have, link it up with that? That's basically
Yeah, we already people have already published some nice papers looking at chromosome scale models for high C, which is like a 3D genome architecture assay. So that's really capturing how the genome folds, which I think is a very important application. The DNA sequence, even though it is a sequence, it's also in fact more like a graph or a complex 3D architecture structure. I think that's very feasible. People already published models on that. We have models upcoming. So I don't think any of these problems are necessarily unsolvable. It's mostly a question of just figuring out the right approach. In most cases, as I mentioned, though, the right approach is not scaling the model up. It's very different. It's like really being aware of the intricacies of the data and figuring out how you can avoid biases and setting up the problem in a way that given the small amount of data you have, you can still really get the best out of the models.
Hi, thanks for the talk. I wanted to clarify a question how you're training the models, particularly in the single cell setting. As I understand that you have some single cell measurement of these molecular profiles across many different cell types, but of course all of these still have the same genetic sequence. So how are you training a model to predict these different profiles from the sequence?
Yeah, you can think of every cell type or every cell as a different task. So the input is the same, but the output is different, right? So you can imagine a multitask model. Then you have millions of thousands of tasks. Each task is a different readout in a different cell type. And your goal is to learn different mappings. You can of course train individual models on each cell or each cell type as well. But each cell is actually not possible because data is too sparse. So the idea is to statistically share information from related cells. You can do it in a dynamic or in a static way. But basically the idea is that even the input is the same, the output is changing by context. And so you're learning a specific model for every context. And the goal really is a prediction in a different context, interpreting the model in that context. So you need a context-specific model for every context.
Thanks for a great talk, Anshul. I had a just slightly detailed question about the simulations where you were moving the transcription factors closer. Is the model in that scenario aware of that distance? And if it is, like, how is it at an intuitive level, at least, squeezing that out of data that are static? And also, how much does it scale combinatorially? Like that was 2. I'm curious if you could comment on those.
Yeah, that's a great question. So that's the reason we apply the 2 different strategies that show. One is the synthetic sequences. And with synthetic sequences, you can easily model distribution. Like that's what you're trying to get at. Is like you can make simulations that have no evidence in the data that was trained on. And so translating gene perturbation. But the key part is the spacing information that I showed. There are enough instances in the genome with sufficient spacing that there are enough statistics for the model to interpolate in that space. So it's really not extrapolating where models typically are not very good at. It's interpolating, it's getting much higher resolution, and more importantly, it's marginalizing the effects of just those two proteins. So if you look at it in the genome, there's lots of things happening at the same time. And so typically people are just marginalized over sequences you observe in the genome, but that is incomplete marginalization. With the simulations, we can really marginalize pretty much everything else out and just focus on those two proteins. And we've done this now for thousands of proteins, and we get really beautiful high resolution sequence maps. Spacing maps, and we've been able to scale this up to higher combinations too. Like we've been able to do 3, 4, looking at effects of density, spacing, orientation simultaneously. How to exactly now make this tractable? Like you can of course simulate, the hell out of the system, but it becomes more and more expensive. We have some tricks and ideas of how to encapsulate all the higher order combinatorics into a single model, like an abstracted distilled model that just captures that. And that could be a powerful sort of engine for the future.
Hi, amazing talk, David Miller. As me as an experimentalist, I find basically the validation with the CRISPR perturbation experiments just fascinating. And I can imagine, you showed a couple of examples where it was very, highly, validating your model. Are there cases that failed in those experiments? And can you extract kind of, features of those genetic variations or sequences or, proteins that might indicate what about the model led to, the experiment not validating it, or something about you mentioned more like holistic chromosomal issues, where the simulation might not just have the scope to do that, to make that prediction.
Yeah, absolutely. So the models do fail miserably a lot of times too. And the key trick is to, our goal is to, we look at observational data firstly. So it's kind of a miracle the model can even make these kinds of causal predictions. It's not entirely a miracle. There are mechanisms of why it's able to do it, but nevertheless, given the data we have, we cannot expect it to be really like an oracle in all possible directions. So the key thing for me is, and for us, is to figure scenarios where we can predict if the model is going to fail. And in many cases, interpretation is very powerful for that. So the model makes lots of false positive predictions in the genome. It's very easy to tell that it's a false positive if you look at the features. Because like usually most real enhancers typically have higher order combinatorics of all of these sequence features. Occasionally the model will find a sequence in the genome and just fire, and there'll just be one DNA word there. So it's just responding to that one word. But that's not usually how most real elements in the genome work. So you can start eliminating a lot of false positives and other kinds of effects through that strategy, right? And there are many other such tricks that allow you to take the model's predictions and then really hone in on things that are really high confidence, and also figure out, what might help you improve predictions next time.
Similar to Libby's.
Yes.
Basically guess which one would be
Exactly.
model well validated.
Exactly. And you can debug the model. So I didn't show you this before. Like if you once we train the models 90% of the features in them were just enzyme bias. And how did we figure that out? The predictions were great, but if you implement the model, look at the features, they make no sense. And they map exactly to the sort of sequence patterns of the enzyme. So you have to build an enzyme model that regresses out enzyme bias and then learns the residual component, which is the biology. Right? So interpretation is not useful just to understand biology, but also debug the model, just as the previous speaker said. Yeah.
Okay, so.
I'm Paul, and this night all the speakers from the morning.